So every conference on lymphoma now actually is quite exciting, and there is so much activity out there. I think the two main areas of activity that I saw were prognostic factors and new drugs. So with prognostic factors, there's a lot about PET scanning, so using PET scanning in different ways that we use it now. We tend to use it now to stage people, so show us where the lymphoma is in somebody's body, and then to assess their response at the end of treatment, and that's a very good use of PET. But actually what people are doing now is using something called metabolic tumour volume, so it's really characterising how much metabolically active lymphoma somebody has at the beginning, and also looking at metabolic heterogeneity, so in other words, are there bits of very active lymphoma and bits of inactive lymphoma, which suggests that there may be cell death going on there. That may be a bad thing. And combining those scores to look at prognosis. So in other words, we do a PET at the beginning, do some clever analysis, and we can then say to people, yes, actually your R-CHOP's going to work well, or your R-CHOP's much less likely going to work well. Now at the moment that's not going to change treatment decisions, but it does mean that we're better identifying high-risk patients to then potentially offer clinical trials to see if we can improve the outcome of high-risk individuals. So I thought that was very interesting, and there was a lot of presentations on that. And then new drugs. Every conference we go on, there's lots of new drugs being mentioned. Very interesting new data on antibody drug conjugates, interesting new data on what's called bispecific antibody therapy. So most antibodies just target one protein on the cell surface, but we're now getting antibodies that target two, and the idea here is they can target two different cells and bring them together, and if you've got a lymphoma cell, it can bring an immune system cell, a T cell, to it and attack it, and we're seeing more and more data on this, which is these drugs are looking very active. Some toxicities, some side effects which need to be thought about, but certainly some activity. And we're seeing much longer follow-up now of some of the earlier new drugs that were brought in a few years ago. So ibrutinib, for example, we're seeing, and some of the other small molecule inhibitors, we're seeing longer-term follow-up in relapsed mantle cell lymphoma, relapsed Waldenstrom's macroglobulinemia, and it's just giving a much better idea of how well these patients do. And I guess the exciting thing is they look good then and they look good now. You know, sometimes a drug that looks good initially doesn't look so good in the long term, but these really are providing robust benefit for patients. So, you know, that's always worth seeing.